A's, T's, G's, and C's, seemingly meaningless letters that could have been picked at random from the alphabet. However, their meaning is much more. These four letters represent the nucleotide building blocks of DNA. Now, I'm sure many of you have learned about these at some point in your education, but what if I told you that the genetic sequence of DNA and RNA could help us to end the pandemic? By utilizing the knowledge of DNA and RNA sequencing, we can develop cures for various diseases and even the disease known as COVID-19 that plagues us now. People are just beginning to learn the possible benefits of DNA sequencing. For example, many of us have gotten vaccines within the last 2 years that were made possible from DNA sequencing. So by informing you about the Moderna DNA sequencing and what that all means, I will explain how it is being used to treat COVID-19 currently, and how it is used to detect other variants. But first, let's talk about what DNA sequencing actually means. So some background information that we need to establish before going into DNA sequencing. DNA is made up of A, T, G, and C. And according to genome.gov, these letters represent nitrogenous bases of nucleotides, which stand for adenine, thymine, guanine, and cytosine, but I would just refer to them as A, T, G, and C to make this simpler. As we look at the diagram behind me, we see that A binds with T, C binds with G, and this goes all throughout the DNA. And these bases are held together through a hydrogen bond. And what's holding these bases into place is the phosphate ribonucleic backbones.
So what differentiates a person from any other living organism, you may ask? Well, every living organism has some combination of A and T and G and C, and we must take into account the complementary base pairing rule, which states that A will bind to T and G will bind to C. Now, this is very important, as you will see in DNA sequencing. According to the DNA Essentials of Molecular Genetics textbook by Gerbach and S. Miglangi, DNA sequencing is defined as a chemical procedure that breaks a terminally labeled DNA partially at each repetition of a base. Now, in layman's terms, sequencing is just basically determining the letter of these, or the order of these letters as they are found in different organisms. Determining the base pairs using the complementary base pairing rule is made more possible due to the next generation sequencers, which is why this is becoming a more prominent area of science. These next generation sequencers go by the name of Illumina, Nanopore, etc., various companies with various names. There's a few more that make the process cheaper, faster, and overall more efficient than ever. Now that we know what DNA sequencing is, let's talk about how it can be used in everyday life. DNA sequencing has been used to treat COVID already, and there is a vast potential for DNA sequencing. According to the Genome Sequencing Technology and Algorithms, DNA sequencing is being used to develop new drugs and therapies for cancer-personalized medicine, and has even been used to develop vaccines. So there is a more widespread and more practical use of DNA sequences than just testing for COVID-19. They are able to combat the long-lasting battle with cancer, and by our understanding of DNA sequencing, we can most likely develop a more efficient treatment course for these patients. According to virological.org, the entire SARS-CoV-2 genome, which is the virus that causes COVID-19, was sequenced by January of 2020. Now, by January of 2020, many of you and I did not know what COVID was, nor have we ever heard about anything about this virus. But it was already sequenced, and this entire genetic sequence, all the A's, the T's, the G's, and the C's, were already posted online for the public access, so companies like Moderna and Pfizer were able to capitalize on this sequence being posted publicly. by reading through the sequence and developing vaccines on it without having the virus anywhere near them. These high-tech companies were able to produce a vaccine that was developed by never coming in contact with it. In addition to DNA sequencing being used to make vaccines, it can also be used to help stop the spread of COVID-19. And here, behind me, is a picture of what the COVID-19 SARS-CoV-2 genome looks like under a microscope. Now, DNA sequencing can effectively end the pandemic by identifying code variants. And these code variants include Omicron and Delta and other ones, and hopefully no future ones. But these variants occur through mutations, and these mutations are alterations in the genetic sequence that happen when the DNA or the gene is replicating. All genes replicate, and this process happens through interphase, the S phase and interphase specifically, in which the cells duplicate, and because of their duplication, their genes and all their genetic information duplicate. Sometimes these replications can be harmful, and they can, as we've seen with Omicron, make the virus, they can change the structure of the virus, make it a little more transmissible, and they can even potentially change the symptoms that occur as a result of getting the virus. According to a scientific journal called Nature, over the last 8 months, over 5 million genomic sequences for COVID-19 have been entered into a sequencing database. This is compared to 1 million genomic sequences that were entered over the first 16 months of the pandemic, so that we can see that sequencing, even over the past couple years, has grown exponentially. According to Cytic Daily, the sequencing of Omicron variants has allowed researchers to find over 30 mutations in the structure of the virus. Now, knowing these mutations can help scientists potentially develop new drugs or determine how to best public health protocols or develop other new therapeutics in the future when new variants ultimately rise, we will see how DNA sequencing works with stopping the spread when the next COVID variant pops up. In the picture behind me here, researchers in a biotech lab are researching the Omicron variant of COVID-19. They've taken different approaches to researching it, such as taking bacterial cultures, as you see the researcher behind me here is, and watching it develop over a period of time in order to understand its structure in comparison to the original variant. An illness that is eerily similar to COVID-19 will be the one caused by the influenza virus, which we are known to as the flu. Now, the COVID-19 virus and the flu virus are eerily similar in their symptoms, however different in their cause. COVID-19 is caused by the coronavirus, which is also known as the SARS-CoV-2 virus, or most commonly known as the SARS-CoV-2 genome. And the flu is caused by the influenza virus. Now, each year emerges a different strain of the influenza virus, which is different strains known as influenza A or influenza B, and they circulate each year. And COVID-19 is similar now, as there are different strains coming up, such as Omicron and Delta, that are emerging and becoming more transmissible each year. In summary, I have explained what DNA sequencing is and how it can be done through various methods, such as Illumina and Nanospore next-generation sequencers. Additionally, I have explained how DNA sequencing is already being used in everyday life, how this technology is being used to try and end the COVID pandemic. Now, despite all great things that DNA sequencing has helped researchers and scientists alike do, there's still a limitless potential of what this technology can do for medicine, for everyone. In the future, thanks to this great scientific feat, the next great cancer treatment or COVID-19 vaccination is just a few A's, T's, G's, and C's away. Thank you very much.